Hello and welcome. This is Bhaskar Napte from Pharma Growth Hub, and in today's video, we are going to talk about five important interview questions related to stability study. So, stability study is equally applicable for research centers as well as manufacturing sites, and this is the questions probably you may have to face during the interviews. Let us talk about the first question. How much excursion in condition for the stability chambers is allowed? Now, this question is related to the excursion possible or allowed into the stability chambers. It can be due to the power failure or breakdown into your stability chambers. But how long the excursion beyond your stability storage condition allowed? For example, if you're loading the sample at long-term conditions at 25 degrees Celsius and 60% relative humidity. If that chamber undergoes breakdown, how long the samples, because the moment the chambers undergoes breakdown, you will be no longer able to maintain the temperature at 25 degrees Celsius and relative humidity at 60% RH. The temperature can go up and the humidity also can get changed. Let us understand, the guidance provided by WHO. So according to WHO, and here is the name of the guideline, that is stability testing of active pharmaceutical ingredients and finished pharmaceutical products. The excursion of 24 hours beyond your set stability condition is allowed. So this one reference can help you out in answering this very important question. And also in case if you are making a SOP for stability study, and if you stuck at how much should be the excursion allowed for the stability conditions for my chamber, now the 24 hours can be the answer for this. But when you look at ICH guideline, that is Q1AR2 now, stability testing of new drug substances and products. So this ICH guideline does not provide any such kind of information onto the allowed excursion. Let us talk about the second important question. And here is the question in front of you. Can be Dosier submitted with 6-month long-term data? So generally, if you look at the ICH guideline or WHO guidelines for stability study, you will find that the dossier can be submitted for your API or for formulation product with 6 months of accelerated and 12 months of long term. Now the question is, if you have only 6 months of long term, can dossier be accepted? Let us understand the guidance provided by WHO. So according to WHO, for existing products that are known to be stable, dossier submitted with 6-month long-term data must get accepted. In case of new drug substances or product, you need to follow the 12-month guideline. That means for new drug substances and new drug products, you must submit the dossier with 12 months of the long-term data. But in case you are not submitting an application for new drug substance or new drug product, in case if your drug substance or drug product is already approved and it is stable, in that situation, your dossier with 6-month accelerated as well as 6 months of long-term data must get accepted. If you look at the ICH guideline Q1AR2, you will not find such kind of a reference onto whether the 6 month of the dossier with 6 month of long-term data gets accepted in case of the earlier approved product which is stable. But when you look at the questions and answers, onto the ANDAs published by FDA, in that answer FDA has clearly stated that yes, in case if you have a dossier with 6 months of accelerated as well as 6 months of long term, that dossier will get accepted. Now here it is very specific to the ANDAs, means what? This is not applicable for the new drug substances and new drug products. So again FDA goes back with the ICH guidelines that the 12 month of long-term data is required for new drug substances and new drug products. It's only for generic products or the products which are already approved in the FDA US region. For those products, the 6 month long-term data, if it's available and if you submit the dossier, it will get accepted. Let us talk about the question number 3 now. Are the accelerated conditions mentioned by WHO and ICH for product to be stored at a refrigerator that is 5 degrees Celsius same? Now the interviewer is trying to understand whether you have read both the guidelines very meticulously. ICH guidelines for the stability study and the WHO guideline for the stability study. So let us understand what are the differences. So there is a little difference when it comes to accelerated conditions for the product to be stored at a refrigerator. Let us understand that very little difference. So according to ICH, that is guideline number Q1AR2, the accelerated condition for a refrigerator product is 25 degree and 60% RH. Whereas if you look at the WHO, the accelerated condition for the product to be stored at a refrigerator condition is 25 degrees Celsius and 60% RH. Now this is the first preference. WHO also talks about you can also select 30 degrees Celsius and 65% RH. The third option, that is 30 degrees Celsius and 75% RH, is also a alternative accelerated conditions for the products which are or which to be stored at refrigerator conditions. I hope you got the difference now in case of ICH and WHO when it comes to accelerated storage conditions for the product to be stored at refrigerator condition. Let us talk about the question number 4. So, how many batches are required for conventional dosage forms with API that is known to be stable? Now, this is again for the not the new drug products. This is applicable for the ANDAs or the products which are already approved. How many batches are required for conventional dosage forms with API that is known to be stable? So, you have a stable API. And in case if you are submitting an application, you must have read that the 3 primary batches are required. Let us understand. According to the ICH, that is guideline number Q1AR2, this guideline does not specifically mention about this particular statement. And hence, you must go with the 3 primary batches. But in case if you look at the WHO has made a very special mention about this particular category, that in case if you are submitting a dossier for conventional dosage forms with the stable API, in that case only 2 primary batches can suffice the requirement. So the 3 batches according to the ICH guideline and 2 batches according to WHO stability guideline. I am talking for the conventional dosage form with the stable API. This is the difference. Let us talk about the last but not the least the questions regarding to the stability study. So for aqueous based product, it can be ophthalmic or injectables. That is aqueous based product. which are packaged in semi-permeable container. Now, what is meant by semi-permeable container? The container which allows the passage of solvent like water or the gases. So in case if you have a aqueous based product which is packaged in a semi-permeable container, does significant change in water loss alone at the accelerated storage condition necessitate testing at the intermediate storage condition? So there is a water loss which is significant in its nature. So for semi-permeable, as I explained at the beginning of this question, that this kind of containers can allow passage of water, and hence for the aqueous based product there can be a possibility of a water loss. And according to the ICH and WHO guideline, the 5% water loss is considered to be a significant change. Now, if you remember that in case of significant change gets occurred at accelerated storage condition, you have to test the product at the intermediate storage condition. Now, the question here is, in case if such kind of significant change in water loss gets occurred at the accelerated storage condition, do you need to test the product at the intermediate storage condition? Now, you may get little confused that why interviewer is asking this question, because it is very obvious, right? In case if the significant change gets occurred, the testing at long-term storage, the testing for intermediate storage condition is very obvious. But before you answer this question, let us understand what is the guidance provided by both ICH and WHO. A significant change in water loss alone at the accelerated storage condition does not necessitate testing at the intermediate storage condition. I am talking in the context of aqueous based product packaged in a semi-permeable container. Isn't it important for you to understand the answer of this question? So water loss alone is not going to trigger a testing at the intermediate storage condition. Of course, in case if there is a impurity failure or potency failure at this accelerated condition, you have to certainly go with the intermediate storage condition. So I hope you must have liked the content, which is very much important when it comes to interview. And in case if you would like to receive such kind of content onto the WhatsApp, it's very important for you to join the Pharma Growth Hub WhatsApp group. So there is a link provided into the description. Click onto the link and join the Pharma Growth Hub. Thank you so much.